very similar to the practice exam, except it might be just a little bit shorter. Okay? Yeah.
Is this gas on?
Oh, okay. So you guys, that's a good question. On the gas valves, as long as the handle is at a right angle, 90-degree angle to the nozzle, it's
Okay.
off.
Okay.
So that's off, but that's also off,
Okay.
as long as they're at right angles. But if you
Okay.
smell
I
gas,
was
let
just
us
looking at
know.
everybody's.
Yeah. If you smell gas, let us know, because they can leak.
Yeah, let's do that.
All right. So let's see here. We got that. We got that. And this is the big one sneaking up on us, that lab practical. So it's not next week, but it's the week after that. And again, folks, we only have 2 lab practicals, and that means there's so much information on each one. So again, don't put off studying. You want to be studying as you go, because it's just a massive amount of information. And then what we're going to do today then, we're starting chapter 19 in the lab manual of fungi, and we do the majority of fungi today, but then next Thursday after the metrics exam, we'll do fungi part 2. And in fungi part 2, we focus on symbiotic relationships of fungi which live with a partner. And we'll talk about lichens, we'll talk about mycorrhiza, and that will also be the lab where we focus on disease-causing fungi. Okay? All right. So fungi, it's a huge topic, you guys. Lots of vocabulary, and your colleagues in the past have said, oh, that was a killer unit to have right before the lab practical, because there's so much vocabulary, so many different ways that fungi reproduce. So I just want to let you all know that there are resources that your colleagues in the past have used successfully. So, of course, here in the lab there's the demos, there's the slides we're going to be looking at. But outside of the lab, your textbook is a wonderful resource. And what I tried to do on your handout down here, I said great resources, Tortori, your textbook, chapter 12, pages 329 to 340. Great descriptions of a lot of the topics we're going to discuss today. Wonderful photographs. Okay. The photo atlas, I put down pages in the 3rd edition, the older photo atlas, pages 159 to 173. If you have the newer edition, just look in the index for the names of the fungi, and it will direct you to the appropriate sections. But again, the photo atlas, you guys, the photos in there are beautiful, well labeled. The lab practical study guide that's posted on D2L, the fungi study guide, I tried to make a table to kind of organize the fungi into their classes. So hopefully even the study guide will be helpful. A lot of folks do Google image searches, so they'll download, they'll print photomicrographs of the fungi, put them on flashcards, and use that as a way to test themselves. Jen, one of your colleagues from a couple of years ago, she sent me all the photographs she had taken in lab of the fungal specimens. So if it's not on D2L, I'll try to make sure to get it up to date. Those are helpful. And then we will be referring to these 3 posters. These posters are really nice study aids. They provide examples of some of the fungi we're going to be looking at today. And these are the 3 major classes of fungi that you will have to know for the lab practical: the zygomycetes, ascomycetes, and the basidiomycetes. So I'll try to refer to the posters as we discuss the different fungi today, but they're really helpful. Okay, so again, folks, what we're going to do, I'm just going to go ahead and just follow the handout. So I'll just try to draw some of the cartoons up on the board. And our challenge today is to describe fungal structure, functions, and classification, and reproduction. So it's a lot of information. So we'll just dive in here. Okay, so the study of fungi is called mycology, and people who make a career of studying fungi are called mycologists. Medical microbiology, medical mycology has become increasingly important as the percentage of immunocompromised people living in our communities have increased. With medical advances, folks that in previous generations might not have survived are now surviving longer. We have an increasing number of organ recipients who are on immunosuppressive drugs. People are living longer. And one disadvantage of living longer is as we age, our immune system starts to lose its efficiency. So we have greater percentages of older folks that are, their natural defenses are somewhat lowered. We have, tragically, we have introduction of HIV/AIDS. The good news is a lot more people that have HIV are living longer lives, okay, but we do have a percentage of our population who's struggling with AIDS, they're immunocompromised. People that are on steroid therapy for chronic inflammatory diseases or autoimmune diseases, all these people we have just described, their natural defenses are somewhat lowered. And most of the fungi, most of the fungal pathogens are opportunistic pathogens. Usually in somebody who's immunocompetent, whose immune system is working well, usually the fungi, they might cause an infection, but usually your immune response is sufficient to keep it localized and controlled. But in folks that are immunocompromised, their defenses cannot keep the fungal pathogens, localized. They can't kill the fungal pathogens. So those are the folks we worry about with the fungi spreading throughout the body. And if you think about it, you already have a patient who's immunocompromised, and now they've been invaded by a eukaryotic pathogen, right? So that often is a greater challenge because you have a eukaryotic pathogen living in a eukaryotic host. And we want to remember, you guys, the more closely related the pathogens are to humans, the harder it is to find drugs that will selectively kill the pathogens without harming our human cells too. Okay, so yeah, these fungal infections, they can be really hard to treat. Okay, now even though we are very interested in pathogenic fungi, we want to recognize most of them are beneficial. Most fungi are free-living, they live out in nature, and one of the really important jobs as nature is that they help break down dead organic matter, dead plants, dead animals, okay? So organisms that live off dead animals or organisms are called saprobes, okay, saprobes. So most fungi are saprobes. They wait until an animal or plant dies, and then they'll digest them, use them as a source of preformed organic molecules. It's relatively few fungi that can't wait and invade us before we're dead. Okay, so, and again, most of them are opportunistic pathogens. Now, one of the challenges in mycology is classification, and we're going to see there's different ways to classify fungi. So we're going to start with kind of the simplest way. We'll look at some structures. We'll move through reproduction, and then we'll talk about the classical way to classify fungi according to sexual reproduction. Okay, so again, you guys, I'm just going to try to follow your handout here. So if we first try to group fungi based on their structure, we've got three basic groups: the yeast, the molds, and the so-called fleshy fungi. Now, yeast are described as unicellular fungi. What does unicellular mean? Well, a single cell, right? Okay. And another characteristic of yeast is that they reproduce asexually by a process called budding. Okay, so today you're going to be looking at some yeast slides, and you'll see these oval yeast cells. Okay, and when yeast are ready to reproduce asexually, meaning just a single parent is going to copy their genetic information and then divide. Okay, so the offspring are genetic clones of their parents. What we'll see is this very cool process called budding. And in budding, we'll have a little extension. It looks like a nose or a little bud that eventually will separate from the parent. We could say this is the parent cell. It will actually separate, grow bigger itself, and then it too can bud. Okay. Now, buds might have different shapes. The yeast we were looking at yesterday, Saccharomyces, the buds look almost like long noses. Kind of cute. But buds may have a more oval appearance. Might be like so. So this process is called budding, and we associate it with yeast. So whenever we look in a microscopic slide and we see cells that are budding, one of the first things that comes to our